Good evening. The other day we discussed the cell wall, cytoplasm, and now we will discuss the remaining cell organelles one by one. Today if I was take the cell organelle, the nucleus is most important. It is the dynamic center of the cell. First organelle, this is discovered by Anton von Leeuwenhoek in RBC. Most details given by Robert Brown in 1831, and Matthias Schleden also gave more details regarding the nucleus. The nucleus is located at the center in meristematic cells, but in peripheral it is positioned in peripheral cells, in permanent cells. See, look here, in a meristematic cell, the prominent nucleus, dense cytoplasm, so the nucleus is at the center. When the cell becomes older, the formation of a central large vacuole and just leave the nucleus aside. It is usually spherical in shape. Usually single nucleus is present in each cell, but rarely you can find two nuclei or multinuclear conditions. As I told earlier, this is the meristematic tissue where you can find the abundant cytoplasm and a prominent nucleus is found exactly at the center of the cell. When the cell becomes older or permanent and formation of a very big large vacuole in the center and leave the nucleus and other parts aside, other floral parts aside. This cell contains the cell organelles. These cell organelles are just pushed aside. There are some cells with binucleate condition, and very rarely you can find multinucleate condition, that is xenocytic condition, in fungi like Rhizopus and some other cells. The nucleus consists of different parts. First one is the nuclear envelope, nucleoplasm, nucleolus, and chromatin reticulum. Now let us see one by one. Nuclear envelope. See, here you can find this is a typical nucleus. There is a nucleolus inside, and this is the nuclear membrane, a double membrane structure. This is called the nuclear membrane, and again you can find nucleolus is there, and nucleoplasm or karyolim found inside, and a thread-like structures you can find inside. That's called the chromatin reticulum. Chromatin reticulum. And here you can have this nuclear membrane, a double membrane structure having some gaps. These are called the nuclear pores. This is called the nuclear pore. So there is a continuation between the nucleoplasm and cytoplasm. The nuclear envelope consists of two membranes with a perinuclear space. That is, the space between two membranes is called the perinuclear space and forms a barrier between nuclear contents and cytoplasm. The outer membrane is continuous with the endoplasmic reticulum. It is continuous with the endoplasmic reticulum and bears ribosomes on it. The outer membrane is continuous, and then you see the outer membrane here, this is inner membrane and this is the outer membrane, and outer membrane is continuous with the endoplasmic, rough endoplasmic reticulum, that is the endoplasmic reticulum having the ribosomes. And interrupted by the minute nuclear pores, I told you already, formed by the fusion of its two membranes. These are passes through RNA and proteins molecules move in both the directions between the nucleus and cytoplasm through the nuclear pores. Next is the nucleoplasm. Is also called the nuclear sap or nuclear matrix or karyoplasm or karyolymph. Just these are synonyms of nucleoplasm, and it contains nucleus, nucleolus, this is very important thing, nucleolus and chromatin reticulum. Various types of proteins, enzymes, nitrogen bases are embedded in the nucleoplasm. nucleolus, deeply stained spherical bodies present in the nucleus. Generally, the nucleus may be one or few present. Few. So, this is first described by Fontana in 1781. The nucleoli is described by the Fontana in 1781. So, the nucleoli may be one to few in each nucleus. The nucleolus is not delimited by any membrane, that you have to remember. Next, nucleolus. This nucleolus, it is contains continuous with the rest of the nucleoplasm and site for active ribosomal RNA synthesis, that is site of nucleolus. The site for formation of ribosomes, that is ribosomal RNA synthesis. It consists of two specific zones, the granular zone and a fibrillar zone. The granular zone contains maturing ribosomes and the fibrillar zone represents the DNA containing genes for ribosomes and ribosomal RNA strand. The nucleolus is concerned with the synthesis of ribosomes. This is very important. The nucleolus is concerned with the synthesis of ribosomes. Next, the chromatin reticulum. It is just a net-like structure found in the nucleus. The DNA containing material in the nucleus that stains deeply with the basic dyes is called the chromatin reticulum. The chromatin reticulum is in the form of long, slender, thread-like structures in the interface nucleus. They entangled together appear as a network called the chromatin network. During cell division, the chromatin threads form rod-like structures called chromosomes. So, in the normal condition, that is interface, it is just like a thread-like structures. At the time of reproductive cell division, they form into threads, form into rod-like structures called the chromosomes. Next, regarding the functions of the nucleus. Regarding the chromosomes, we will have another chapter later. Now, let us see the functions of nucleus. Nucleus is the dynamic center of the cell. It controls all the cellular metabolic activities. Therefore, the nucleus is essential for all living cells. The nucleus initiates cell division, another important point, and nucleus is responsible for heredity. It helps in the transmission of characters from parents to offsprings through the sex cells. So, that is, in sex chromosomes, we can find the hereditary characters are just like the sex characters or the genetic characters are passed from one generation to another generation. Right. Next. So, the next cell organelle is another very important one because of the plants. They are having the capacity to carry the photosynthesis. So, this is very important. The plastids, unique character in plants, they can found only in plants only, not in any other organisms. They are concerned with synthesis of storage of food materials. The term plastid was coined by Haeckel in 1866. They are classified into three types by Schimper. Schimper, Schimper in 1883. Schimper, Schimper. They are leucoplasts, chromoplasts and chloroplasts. So, these three named by Schimper only. Now, let us see, this is a diagram shows different types of chloroplasts, these plastids. The primitive, that is a beginning, is a proplastid from which they are originated different types of plastids. Leucoplasts, these are the colorless plastids. Chloroplasts are green colored plastids and chromoplasts are red and yellow colored plastids. Again, these leucoplasts depend upon the storage. Amyloplast, which store the starch, eleuroplast, which store fat, and eleuroplast, it stores the proteins. So, these are the different types of plastids and their function. Now, let us see, the first one is the leucoplast. These are the colorless, without any pigment. They are of various sizes and shapes with the stored nutrients. Amyloplasts, they store the starch. Ureoplasts, oil and fat. Eleuroplasts, proteins. Found in underground organs. Generally, these leucoplasts are found in the underground organs and parts kept away from sunlight. Away from sunlight. Next is the chromoplast. Chromo means colored plastids. Colored due to the fat soluble carotenoid pigments like carotenes and xanthophylls. The carotenes are red in color, xanthophylls are yellow colored pigments. Generally they are found in the pericarp of fruits. Different color of fruit due to these chromoplasts only. And pericarp of the fruits, that is outer wall of the fruit, and in petals they have different colors, that is due to the presence of chromoplasts. So because of this colored pigments, colored fruits and colored the flowers, generally the pollination by insects and seed dispersal by agents, different agents takes place, especially the insects and animals due to this color. Next, chloroplasts. These are the green plastids found in leaves and stem. They are concerned with photosynthesis. They are large, lens-shaped, oval or spherical, discoid or even flat or ribbon-shaped. The number varies from 1 to many. One chloroplast found in Chlamydomonas, near to 20 to 40 per cell in palisade cells. You can find the number of chloroplasts. It consists of an envelope. The chloroplast, that's the structure of the chloroplast. It consists of an envelope, the stroma and lamellar systems. A cover, outer cover, and inside is called stroma, and inside you can have a number of lamellar structures. So envelope, let us see the envelope. It is double membranous, and stroma, that is inside, filled with fluid, and lamellar system, and also having circular DNA, 70S type of ribosomes and various types of enzymes. So here it is having a 70S type of ribosomes. This is the characteristic feature of the prokaryotes. And also the circular DNA, that is also character of prokaryotes. So the prokaryotes found in eukaryotes. That is a very interesting thing here in ribosomes, in chloroplast as well as in mitochondria. There you can find the circular DNA and 70S type of ribosomes, and they are semi-autonomous. This is given semi-autonomous. They also have a role in the transfer of characters from one generation to another generation. So not only the nucleus but also these plastids and mitochondria also have a role in the transfer of characters because of having DNA, protons, this ribosomes, etc. They are capable of producing the genes, just like the genes. Next we have the structure of a chloroplast. I told you already, this is a semi-permeable, outside the membrane bound structure, double membrane. Each membrane is similar to the structure of a plasma membrane, and inside you can find the stroma. Inside part is known as the stroma. In this stroma, you can find a bunch like a bundle like structures, or a number of thylakoids are there. These are all thylakoids. They are arranged, superimposed, a coin like one by one like that. Each one is called a thylakoid. These thylakoids are connected by stromal lamellae, and inside you can have the stroma. So here another diagram is given. The thylakoids inside the stroma, thylakoids is just like a plate-like structures having in between space is found. All the photosynthetic pigments are just located in the thylakoids only. So next we will see lamellar structure consists of grana, contain flattened disc-like structures, just to have seen. Each disc-like structure of granum is called thylakoid. The name is given by Menka in 1961. There are about 40 to 100 grana in each chloroplast. Each granum consists of 20 to 50 thylakoids. Various kinds of pigments like chlorophyll A, chlorophyll B, carotenoids, xanthophylls, etc. arranged in the lamellae. Thylakoid membrane consists green pigments abundantly and other colored pigments and enzymes that absorb light and generate ATP and NADPH2 during the light reaction. NADH2. Here you note down NADH2. These are formed during light reaction. I think you know the photosynthesis is mainly divided into two reactions. One is called the light reaction, another is called the dark reaction. In the light reaction, the photosynthetic pigments absorb light and prepare ATP and NADPH2. The ATP and NADPH2 are used by enzymes in stroma to convert CO2 into sugars. This is the second dark reaction. In the dark reaction, the light reaction takes place in the grana, dark reaction takes place in the stroma. In the stroma, the ATP and NADPH are used by enzymes in stroma to convert carbon dioxide into carbohydrates. So, in the photosynthesis, there are two reactions. One is granum, that is light reaction. Another is called dark reaction that takes place in the stroma of the chloroplast. Regarding the functions, chloroplasts prepare food material by photosynthesis. The light phase reactions of photosynthesis takes place in the granum, as I told you already, and produce ATP and NADPH2. The first one, the light phase. In the light phase of the reactions, they use the photosynthesis. They use this light energy and prepare ATP and NADPH2, and oxygen is released in the first phase, the light reaction. So, just using the sunlight and water, prepare energy and release of oxygen. In the second phase, they utilize carbon dioxide, reduce carbon dioxide with the help of ATP and NADPH2 that are formed in the light reaction and convert into sugars. So, dark phase reactions occur in the stroma region. So, that is about this chloroplast. And the next one is the mitochondria. This is another very essential. It is found both plants and animals. Especially in the eukaryotic organisms, you can find mitochondria. And these mitochondria are one of the most important cell organelles found in the cytoplasm of eukaryotic cells. They are mainly concerned with the respiration. So, transduction of energy from food materials to ATP. So, they oxidize, the oxidation of food materials into energy. So, in that process, carbon dioxide is released. So, generally people used to tell the respiration is taking oxygen inside and release carbon dioxide outside. That is not at all a respiration. That is simply a breathing. Actual respiration is the oxidation of food materials. That is called respiratory substrates, and release energy in the form of ATP. In the meanwhile, carbon dioxide is released as a byproduct. So, that is the actual respiration. So, they were first described, the mitochondria were first described by Altman in 1894 under the name bioplast. At the time, he gave the mitochondria as a bioplasts. Later, the mitochondria name was given by Benda in 1898. After 4 years, the name is changed, the bioplast name is changed to mitochondria. Regarding the morphology, the shape, size and number of mitochondria are variable from cell to cell. Mitochondria may be spherical, oval, cylindrical, etc., but usually filamentous or granular. The diameter is also about from 0.5 microns and this millimicrons, and the length is variable, reaching a maximum size of 7 millimicrons. Usually plant cells contain fewer mitochondria than the animal cells of the same size, but in animals you can find more mitochondria when compared to plants. By using Jensen green be a strain of mitochondria can be observed in living cells. This can be seen by using this strain. So here the structure, a mitochondria is just a longitudinal section is given, a mitochondria, where you can find outer membrane. There are two membranes, outer membrane and inner membrane having infoldings. These are called the cristae, and inside the portion is called the matrix. And here you can find a DNA. As I told you, a circular DNA is here, and also 70S type ribosomes also found inside. Regarding this membrane, outer membrane, inner membrane, there is a space that I will tell you later. This mitochondria having a outer mitochondrial membrane, outer membrane, inner membrane, in between there is a space. There is a space, that is outer membrane, inner membrane, there is a space that is called perimitochondrial space, and inner membrane, and cristae formed by the infoldings of the inner membrane, and inside is called the matrix. So mainly the membrane and the cristae and the matrix. These are the three parts. The outer membrane forms boundary and composed of lipids and proteins, equal ratio of proteins and lipids regarding the outer layer. Whereas the inner layer, the combination changes lipid and proteins in the ratio of 20:80, forms a number of infoldings. Those are called the cristae. Why this cristae help in the increase of the enzymic action of the matrix or inner compartments. That is due to this cristae increasing the surface area for enzyme actions into the matrix. Both membranes have specific enzymes associated with aerobic respiration. Inner membrane, here inner membrane, inner foldings have got a fixing of F1 particles, stocked particles are there. Here you can find this is a F1 particle projecting into the matrix. Each F1 particle has three parts. Inside the cristae, as I told you already, there you can find this is the outer membrane, and inner membrane having infoldings. These infoldings are called the cristae. If you enlarge one infolding, this is like this. There you can find a number of F1 particles. So each F1 particle having a base, a stalk, and a head. In the head contains of the ATP synthetase enzyme. So inner membrane of the cristae is lined with numerous stalked particles. Those are called the F1 particles or elementary particles, project into the matrix. Each F1 particle has three parts: head piece, and stalk and base. So base, stalk, and head. Enzymes and coenzymes of electron transport chain occur in cristae membrane where F1 particles are present. They act as electron acceptors in reactions of oxidative phosphorylation. So here you should know something regarding the respiration. As in this photosynthesis, there are two reactions, light reaction and dark reaction. In respiration, you have four reactions. The first one is the glycolysis. That glycolysis is nothing but the conversion of one molecule of sugar into two molecules of pyruvic acid. It takes place in the cytoplasm, and then this pyruvic acid enter into the mitochondria. In the mitochondria, there you can find the Krebs cycle is there. There you can find a number of NADPH2 and FADH2 are formed. These forms enter into the cytochrome system that you can find in F1 particles. In F1 particles, they become the reduced particles become what happen? Oxidized and formation of ATP. So oxidation and formation of ATP. So this is called oxidative phosphorylation. So ATP are formed in aerobic respiration. So that's why in respiration, the mitochondria and the cytoplasm only glycolysis, remaining total release of energy etc. takes place in the mitochondria only. So that process, the electron acceptor reactions are called oxidative phosphorylation. Phosphorylation means the creation or formation of ATP by oxidation, oxidative phosphorylation. So the same in photosynthesis, they also formed this ATP, that is called oxidative photophosphorylation. That is phosphorylation in the presence of sunlight, photophosphorylation. Here oxidative phosphorylation. So the matrix possesses single circular DNA molecule, a few tRNA molecules, ribosomes of 70S, and enzymes required for functioning of the this is very important, mitochondrial genes. As I told you already, they are the chloroplast genes, these are mitochondrial genes, so that these are also semi-autonomous. They also carry some characters from one generation to another generation, and preparation of proteins also by these things. That's why they are called semi-autonomous bodies. So in a cell, in addition to nucleus, the chloroplast and mitochondria are semi-autonomous bodies. In eukaryotic cells, the first phase of respiration is glycolysis. I told you already, it takes place in the cytoplasm. While the second phase, Krebs cycle or tricarboxylic acid cycle or citric acid cycle or aerobic cycle, is carried out by enzymes in mitochondrial matrix and cristae. Glucose breaks into carbon dioxide and H2O takes place in cytoplasm and mitochondria. Cytoplasm first reaction and the remaining in mitochondria. The complete aerobic degradation of glucose molecule to carbon dioxide and H2O is coupled to synthesize 38 molecules of ATP. So one glucose molecule gives 38 ATP molecules for energy. These ATP molecules break, their phosphate bonds break when hydrolyzed, they break and give energy. That energy is used to carry all the biological activities. Regarding the functions, the cristae are the sites of oxidative photophosphorylation, so oxidative phosphorylation and electron transport. It produces energy currency of the cell, ATP. So generally ATP is the energy currency, and those are present in the mitochondria. So these are called the powerhouses of the cell. Powerhouses of the cell. Energy currency of the cell is ATP, and powerhouses of the cell is the mitochondria. So the mitochondria is very essential for this respiration. So thank you. Tomorrow we will take some more cell organelles.